press the bell icon on the YouTube app and never miss another update.
Namaste children. In our previous class we discussed about synapse and types of synapses. So synapse is nothing but it is a functional junction between two neurons. That means it includes post synaptic membrane, cleft synaptic cleft, and so synapse includes presynaptic membrane of axon and synaptic cleft. It is a junction between presynaptic membrane and postsynaptic membrane, and postsynaptic membrane is seen on dendron. This is about synapse. It is a functional gap between two neurons, that between the axon and dendron. So we discussed two types of synapses, that is chemical synapses and electrical synapses. So chemical synapses alli enagutte neurotransmitters are present, whereas in electrical synapses neurotransmitters are absent. Then in the chemical synapse we see delay in the conduction, whereas in electrical we see faster rate of conduction. Again, the gap is very less. The gap synaptic cleft is 10 to 20 nanometer in chemical, whereas in electrical it is just 0.2 nanometer. And chemical is always unidirectional from axon to dendron. Electrical is both, we can see, bidirectional from axon to dendron and also from dendron to axon. But in higher vertebrates we see chemical synapses only. In very few area we see electrical synapses. This electrical synapses is seen in invertebrates. So that's the difference between chemical and electrical synapses. So today we will discuss properties of neuron. So first properties of neuron, electrical potential. So due to this presence of electrical potential, neurons are excitable and conductible. So as all the other cells, even nerve cells is surrounded by extracellular fluid which contain ions and salts. So this electrical potential in the membrane of the nerve is maintained by usually the extracellular fluid, that means the fluid present outside the nerve cell is rich in extracellular fluid. We see sodium ions, whereas in exoplasm it is rich in potassium and negatively charged protein molecules. Outside the nerve fiber, that is in extracellular fluid, that fluid is rich in sodium ions. Inside the nerve fiber, that is inside the exoplasm or neuroplasm, they are rich in potassium and negatively charged proteins. Nerves at rest, they have positive charges outside and negative charges inside. This is mainly maintained by sodium potassium pump. So it is an active transport. It is an active transport which allows 3 sodium to move out, efflux of 3 sodium into extracellular fluid from the exoplasm and 2 potassium ions, influx of potassium ions into exoplasm. So, this again helps to maintain positive charges outside and negative charges inside. And one more is axolemma or neurolemma is permeable to sodium ions and impermeable to potassium ions and negatively charged protein molecules. So, this permeability of membrane again helps to maintain positive charges outside the axolemma and negative charges inside the axolemma. So, this helps to maintain negative charges inside and positive charges outside. That means the nerve fiber will be in a polarized state. Nerve fiber will be in a polarized state. This difference in the charges around the axolemma is nothing but electrical potential. Due to this electrical potential, the nerves or neurons are excitable or unexcitable and also conductable. When we apply stimulus, we see reversal of polarity that helps in generation of action potential, and that action potential again, because adjacent area ಏನಾಗಿರುತ್ತೆ, this area stimulus apply ಮಾಡಿದಾಗ applied area depolarize ಆಗಿರುತ್ತೆ, adjacent area again polarized ಇರುತ್ತೆ. Electrical circuit will develop between those two and impulse will be carried till the axon terminal, and from axon terminals to impulse will be transferred to other neurons through a gap called synapses. That all we discussed in our previous class. So, this is one property of nerve fiber or neurons. Idu nimge in your textbook they are not given, but tilkondirbeku because they may ask in your NEET exams. Second property, threshold level. Threshold level andre, to generate action potential, the stimulus should have a certain minimum strength. Suppose idu chalk, just ನಾನು ಸುಮ್ಮನೆ ಸ್ವಲ್ಪ ಪ್ರೆಷರ್ ಹಾಕಿದರೆ ಬೆಂಡ್ ಆಗಲ್ಲ, ಅಂದರೆ ಜಾಸ್ತಿ ಪ್ರೆಷರ್ ಹಾಕಿದರೆ ಏನಾಗುತ್ತೆ, ಒಂದು ಮಿನಿಮಮ್ ಪ್ರೆಷರ್ ಹಾಕಿದರೆ it is going to bend. Like that, the stimulus should have certain minimum strength to excite neurons. So, weaker stimulus will not able to excite neurons. The stimulus should have certain minimum strength to excite neurons. That strength is called threshold level. That strength is called threshold level. Majority of the neurons have threshold level between 5 to 15 millivolts. The next one is all or none phenomenon. All or none phenomenon means to generate action potential, the stimulus should have a certain minimum strength. Below that strength, the nerves are not excitable. So, the nerves are excitable only at the threshold level, not below that. That is nothing but all or none phenomenon. So, all or none phenomena means neurons are able to generate action potential or excitable only at threshold level not below that. So, neurons are able to generate action potential or excitable only at threshold level and above. Threshold level and only at threshold level or above, not below that. So, that is nothing but all a non-phenomenon. There is no weak stimulus or partial stimulus. So, neurons are able to generate action potential only at their threshold level or above, not below that. That is nothing but all a non-phenomenon. Next property of neuron. Unidirectional. Always impulse is carried in one direction only, that is from axon to dendron or dendrites, axon to dendrites. Next, insulation. Insulation means the ions are not able to leak from the neurons because they are insulated by myelin sheath. They are insulated by myelin sheath. So, unidirectional, always the impulse is move from axon to dendron. It is follows forward movement. Insulation is provided by myelin sheath. This myelin sheath around the axonema acts as an insulator that prevents the leakage of ions. That leads to faster rate of conduction. So, the rate of conduction is faster in myelinated neurons when compared to non-myelinated neurons. So, ನೀವು ಬರಿಬೋದು faster rate of conduction impulse is conducted faster in myelinated neurons and compared to non-myelinated neurons. So, ಇಲ್ಲಿ see, this myelin sheath is interrupted by squamous cells. They are interrupted by squamous cells. This is cell body, dendrons, axon terminal. The myelin sheath is present around the axolemma that helps in faster rate of conduction of impulse. And this myelin sheath is interrupted by schwann cells. So, it is interrupted by schwann cells, and the junction between schwann cells is called nodes of Ranvier. It is called nodes of Ranvier. ಇಲ್ಲಿ ನಾವು saltatory conduction ಅಂತ, saltatory type of conduction of impulse is seen in myelinated neuron. Saltatory type of impulse conduction is seen in myelinated neuron. What is saltatory type of impulse conduction? So, illenagutte, the axolemma, as the axolemma is surrounded by myelin sheath, it is not direct contact with the extracellular fluid. So, the nodes of Ranvier alli there is absence of myelin sheath. So, illenagutte, axolemma will be in contact with the extracellular fluid. So, the action potential is going to be generated in the nodes of Ranvier, not in the axolemma which is surrounded by myelin sheath. So, illinda when stimulus is applied, action potential is generated here. From there it transfers to axon. Axon illige bartidankala enagutte, from here to the stimulus will shift to this node of Ranvier. From here to again it shifts to here, then it shifts to nerve terminal. So, the action potential shifts from one nodes of Ranvier to another nodes of Ranvier, because this part is not in contact with the extracellular fluid. Only in the nodes of Ranvier the axolemma is in contact with the extracellular fluid. There we see movement of ions, movement of sodium ions into the exoplasm. Alli generation of action potential we are able to see. So, hagagi enagutte, the impulse will jump from one nodes of Ranvier to another nodes of Ranvier. Hagagi the rate of impulse conduction is more in the myelinated neuron when compared to non-myelinated neuron. This jumping of action potential from one nodes of Ranvier to another nodes of Ranvier is called saltatory type of impulse conduction. Ade neevu non-myelinated neuron alli enagutte. So, this is cell body, here the impulse has to illi action illi stimulus apply madirtira neevu, then action potential will be generated here. Then the area, this part area will become depolarized and next part will be in a polarized state. So, it will shift from here to here. Then this area becomes repolarized, this area becomes depolarized. Again, jumping of action potential jumping of impulse does not take place in non-myelinated neurons. Throughout the axon, the impulse, now in previous class I discuss madidvala, it shifts from this area to this area, this area to this area. When this area becomes depolarized, adjacent area will be in a polarized state. Then stimulus will shift to B region. Then from B to C, C to D, like that it has to reach nerve terminals, that's axon terminals. Hagagi there is delay in conduction of impulse in non-myelinated neurons, in non-myelinated neurons. Suppose ಇದಕ್ಕೆ ನಾವು ಹೆಂಗೆ ತಿಳ್ಕೊಬೋದಂದ್ರೆ, ಮೈಲಿನೇಟೆಡ್ ಅಲ್ಲಿ ಏನಾಗುತ್ತೆ, ನೀವು ಇಲ್ಲೊಬ್ರು ನಿಲ್ಲಿಸಿರ್ತೀರಾ, ಫಸ್ಟ್ ಪರ್ಸನ್, ಇಲ್ಲಿ ಸೆಕೆಂಡ್ ಪರ್ಸನ್, ಇಲ್ಲಿ ಥರ್ಡ್ ಪರ್ಸನ್. ಸೊ ನೀವು ಬಾಲನ್ನು ಇವ್ರಿಗೆ ಕ್ಯಾಚ್ ಮಾಡ್ತೀರಾ, ಇವ್ರಿಗೆ ಸೆಕೆಂಡ್ ಪರ್ಸನಿಗೆ ಕ್ಯಾಚ್ ಆಗ್ತಾರೆ, ದೆನ್ ಸೆಕೆಂಡ್ ಪರ್ಸನ್ ವಿಲ್ ಥ್ರೋ ದಟ್ ಟು ದ ಥರ್ಡ್ ಪರ್ಸನ್. ಸೊ ಬಾಲ್ ಫಾಸ್ಟಾಗಿ ಥರ್ಡ್ ಪರ್ಸನಿಗೆ ರೀಚ್ ಆಗುತ್ತೆ. ಅದೇ ನೀವು ಇಲ್ಲಿ ಯಾರೂ ಇಲ್ಲದೇ ನೀವೇ ನಡ್ಕೊಂಡು ಹೋಗಿ ಇವ್ರಿಗೆ ಕೊಡಬೇಕು ಅಂತ ಅಂದರೆ ಇಟ್ ವಿಲ್ ಟೇಕ್ ಟೈಮ್. ಸೊ ದ್ಯಾಟ್ ಹ್ಯಾಪನ್ಸ್ ಇನ್ ನಾನ್ ಮೈಲಿನೇಟೆಡ್. ಸೊ ಮೈಲಿನೇಟೆಡ್ ಏನೇನಾಗುತ್ತೆ, ಜಂಪಿಂಗ್ ಆಫ್ ಆಕ್ಷನ್ ಪೊಟೆನ್ಷಿಯಲ್ ಆರ್ ಜಂಪಿಂಗ್ ಆಫ್ nerve impulse takes place from one nodes of Ranvier to another nodes of Ranvier. That mainly helps in faster rate of conduction. So that is the difference between conduction between myelinated and non-myelinated nerve fibers or neurons. ನೀವು ತಿಳ್ಕೊಬೇಕಾಗಿರೋದಿಷ್ಟೇ, faster rate of impulse conduction is seen in myelinated neurons and delay in conduction seen in non-myelinated neurons. Saltatory type of nerve conduction is seen in myelinated neuron and it is not seen in non-myelinated neuron. ಮತ್ತೆ ಕೇಳ್ತಾರೆ, what is saltatory type of conduction? So there is jumping of nerve impulse or action potential from one nodes of Ranvier to another nodes of Ranvier. That is nothing but saltatory type of nerve conduction. So that is about insulation and conduction. Then refractory period. These all points you have to remember for your competitive exams. The next we have refractory period. Refractory period means once the impulse is generated, so this is, so here negative, outer positive, so when we apply stimulus for the first time, when we apply stimulus for the first time, this area become depolarized and outer will become, so inner become positive, outer become negative. So this area will become depolarized and action potential is generated here. After that it starts shift from A to B, B to C, like that till it reaches axon terminals. This is axon terminals. When we apply stimulus for the first time, action potential will be generated here and it will be carried to the axon terminal. Immediately the nerve will undergo rest. After conduction of impulse, the nerve will go a period of rest called refractory period. After, when we apply stimulus, the action potential is going to be generated and it is conducted along the length of the axon till it reaches axon terminals. Immediately after that, the nerve fiber will go a period of rest, that is called refractory period. During that refractory period, if you apply stimulus, second stimulus, then there is no nerves will not able to excite or conduct. When we apply a stimulus, the nerves will generate action potential and it is conducted and it is carried along the length of the axon till they reach axon terminals. After that, the nerve fibers undergo a period of rest. That rest period is called refractory period. When you apply stimulus in refractory period, when we apply stimulus in the refractory period, nerves are not excitable or conductible. Neurons are not excitable and conductible. So that is nothing but refractory period. So when we apply stimulus for the first time, they are able to excite and conduct the impulse, but then after that they will go refractory period. During refractory period, if we apply stimulus, whatever the strength of the stimulus, neurons are not able to excite or conductible. That is about the refractory period. And the last property of neuron is impulse, nature of impulse. Nature of impulse. See, if you stimulus, when we take food from the tongue, the information has to carry to the brain. In the same way, how the action potential is generated and carried to the axon terminal, from there it will be carried to the brain. So, whatever may be the stimulus, the carrying of information to the brain is in the same way, that is generation of action potential, and it is conducted along the length of the axon, then it shifts to next neuron, like that it reaches the brain. So, impulse tongue-ಇಂದ ಬ್ರೈನಿಗೆ ಇನ್ಫಾರ್ಮೇಷನ್ ರೀಚ್ ಆಗೋದು ಥ್ರೂ ಅ ಸೇಮ್ ವೇ, ಆ್ಯಂಡ್ ಫ್ರಮ್ ವಾಟ್ ವಿ ಸಿ, ವಿಷನ್ ಆರ್ ವಾಟ್ ವಿ ಹಿಯರ್, listening, everything, information carried to the brain will be in the same way. The nature of impulse carried by the different sense organs to the brain will be the same. ಎಲ್ಲಿ ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ಆಗುತ್ತೆ ಅಂದರೆ ರಿಜಿಸ್ಟರಿಂಗ್ ಡಿಫ್ರೆಂಟ್ ಇನ್ಫಾರ್ಮೇಷನ್, ರಿಜಿಸ್ಟರಿಂಗ್ ಸೆನ್ಸೇಷನ್ ಇನ್ ದ ಬ್ರೈನ್ ಡಿಪೆಂಡ್ಸ್ ಅಪಾನ್ ದ ಡಿಫ್ರೆಂಟ್ ಪಾರ್ಟ್ಸ್ ಆಫ್ ದ ಬ್ರೈನ್ ವೇರ್ ಇಟ್ ರಿಸೀವ್ಸ್ ಇಟ್. ಬ್ರೈನಲ್ಲಿ ಆಲ್ಫ್ಯಾಕ್ಟರಿ ಏರಿಯಾ, ವಿಷನ್, ಆಡಿಯೋ ಏರಿಯಾ, ಡಿಫ್ರೆಂಟ್ ಏರಿಯಾಸ್, ಮೆಮೊರಿ, ಹಿಪ್ಪೋಕ್ಯಾಂಪಸ್ ರೀಜನ್, ಹೀಗೆ ಡಿಫ್ರೆಂಟ್ ರೀಜನ್ಸ್ ಇರುತ್ತೆ. ಅಲ್ಲಿ ಅದು ರಿಸೀವ್ ಆದಾಗ ದೆನ್ ವಿ ಆರ್ ಏಬಲ್ ಟು ಡಿಫ್ರೆನ್ಶಿಯೇಟ್ ದ information. We are able to differentiate information. So, nature of impulse, the impulse carrying from sense organs to brain is same. All the impulse from the different sense organs is carried in the same way to the brain. It is going to differentiate from where the part of the brain which receives it. It is going to differentiate, or it is going to register in the brain. It is mainly based on different parts of the brain where it receives it. That is about nature of impulse. So, these are all properties of neurons. Then we will discuss about meninges. So, we know that central nervous system consists of brain and spinal cord. Brain is protected in the brain box called cranium. Spinal cord is protected in the vertebral column. They are protected in the vertebral column, and this brain and spinal cords are also protected by meninges. They are also protected by meninges. Meninges are there nothing but protective cover. Protective covering membranes of brain and spinal cord. So, protective covering membranes of brain and spinal cord are called meninges. So, we have three types of meninges. One is dura mater, another one is arachnoid mater, another one is pia mater. We have three meninges which supplement the function of bones in protecting the brain and spinal cord. So we have three meninges: dura mater, arachnoid mater and pia mater. Skull, dura mater, subdural space, arachnoid matter, sub and yellow one is pia mater. This is meninges of brain and spinal cord. So we have three meninges: dura mater, pia mater, dura mater, arachnoid mater and pia mater. Dura means tough, mater means mother. So dura mater, it is the outermost covering of brain and spinal cord, which is tough and it is adhered to skull. It is adhered or it is attached to the skull. Below the skull, the first layer of meninges is seen, that is dura mater, which is non-vascularized and tough. It is tough and non-vascularized. Below the dura mater, we see a space that is called subdural space. Sub andre kelgade. So the space below the dura mater is called subdural space. Below that we see a second meningeal layer called arachnoid matter. Arachne means spider. Arachne means spider. Again, matter means mother. This arachnoid matter, below that arachnoid matter we see subarachnoid space where we see presence of cerebrospinal fluid. Cerebrospinal fluid is present in subarachnoid space. And below that we see a third layer of meninges, that is called pia mater. Pia means tender. Pia means tender. In Latin, pia means tender, and again mater means mother. So the innermost layer is pia mater, which is attached to the brain. This part is brain. It is attached to brain. Understood? So dura mater, arachnoid matter and pia mater. The space between dura mater and arachnoid matter is called subdural space. The space between arachnoid matter and pia matter is called subarachnoid space. Nimi competitive keluvantadu. Dura mater and arachnoid both are avascular. Where avascular means that does not supply with blood, whereas pia mater is vascular. It is vascular and attached to that meningeal layer is attached to brain and spinal cord, and cerebrospinal fluid is present in subarachnoid space, produced by choroid collection of capillaries called choroid plexus, which extend from pia mater to subarachnoid space. This cerebrospinal fluid is produced by choroid plexus, nothing but collection of capillaries. So what is the function of cerebrospinal fluid? It acts as a shock absorber. It prevents the friction of brain and spinal cord against the bones. Cerebrospinal fluid it acts as a shock absorber. It prevents the friction of brain and spinal cord against the bones, and also it provides a medium for the exchange of food materials, respiratory gases and excretory products, all exchanged between the blood vessels and nervous tissue through this cerebrospinal fluid. Cerebrospinal fluid. Nearly 150 ml of fluid is present in both male and female. This cerebrospinal fluid is produced by choroid plexus present in the subarachnoid space, acts as a shock absorber, prevents the friction of brain and spinal cord against the bones, and also it provides a medium for the exchange of food materials and excretory products and other substances between the blood and the nerve cells. Next we will go with and into ನೆನಪಿಟ್ಕೊಳ್ಳಿ dura mater, arachnoid are avascular, whereas pia mater is vascular. Dura mater is attached to the skull and vertebrae, whereas pia mater is attached to the brain and spinal cord. And we see the grey matter and white matter. The grey and white matter are the tissues present in central nervous system. ನೋಡಿ ಇಲ್ಲಿ ಬರ್ದಿದ್ದೀನಿ ನಾನು, ಇದು ಬ್ರೈನ್ ಆಗಿರೋದ್ರಿಂದ ಔಟರ್ ಇಸ್ ಗ್ರೇ ಮ್ಯಾಟರ್, ಇನ್ನರ್ ಇಸ್ ವೈಟ್ ಮ್ಯಾಟರ್. ಸೊ ವಿ ಹ್ಯಾವ್ gray and white matter. They are the tissues present, they are part of central nervous system, differentiated into grey or white matter depends on the part of nerve cells involved, depend upon the parts of nerve cells involved. The grey matter, you see cytons and dendrites. Cytons and dendrites. In white matter, we see myelinated neurons. We see myelinated neurons. So, in the grey matter, we see cytons and dendrites. In white matter, we see myelinated neurons. ನೀವು ನೆನ್ಪು ಮಾಡ್ಕೋಬೇಕಾಗಿರೋದು ಏನಂದರೆ in brain outer, in cerebral cortex we see grey matter and inner we see white matter. So, brain alli outer part cerebral cortex alli we see grey matter, that means they contain cytons of cell body and dendrites of nerve cell. White matter contains myelinated part, myelinated neurons. Spinal cord alli ulta agutte, spinal cord alli outer is white matter, inner is grey matter. Understood? So, in brain outer is grey matter whereas inner is white. In spinal cord you see outer is white, it consists of white matter, inner is grey matter. That means outer side of the spinal cord we see white matter, means they contain myelinated neurons. Grey matter contains cell bodies and dendrites. Idu ulta agutte, idu neevu competitive ನೆನಪಿಟ್ಕೋಬೇಕು. In brain outer contain grey matter, inner contain white matter, whereas in spinal cord outer contain white matter, inner contain grey matter. This is about the grey and white matter, and also we discussed about meninges and how they are going to protect brain and spinal cord. So, ಇಲ್ಲಿ meninges ಅಲ್ಲಿ ನೀವು ನೆನಪು ಮಾಡ್ಕೋಬೇಕಾಗಿರೋದು what are meninges ಅಂತ ಕೇಳ್ತಾರೆ, and mention the different types of meninges. Meninges are protective coverings of brain and spinal cord. We have three types of meninges: neuron matter, arachnoid matter and pia-matter. So, cerebrospinal fluid is present in subarachnoid space, which acts as a shock absorber and it prevents the friction of brain and spinal cord against the bones, and also it provides a medium for the exchange of food material and other respiratory gases between the blood and nerve cells. Then white and grey matter bandaga, grey contains cytons and dendrites, white matter contains myelinated neurons. In brain outer part is grey and inner part is white. In spinal cord outer is white and inner contain grey matter. So, that is about meninges. In the next class we will discuss about central nervous system. Thank you.